Everyone, let me get started here. I'm just pulling up this presentation. All right, hello and welcome to today's session. Have we started recording?
Yes, Joe, it is on.
Okay, great. Hello and welcome to today's session on bioinformatics for precision oncology. Today we're going to be discussing big data and how it changes our approach to cancer research, as well as translation to clinical practice. We will speak about the challenges to traditional hypothesis-first approaches, the relationship between statistical methods, and then machine learning. We're also going to go over several sections in the T-BioInfo Platform, which was built for accommodating both traditional bioinformatics for processing data, as well as big data mining and machine learning that is becoming more commonplace in big data studies. First, let's go over the course progress and see how you are getting along with the online materials. We will test how familiar you are with the process by asking a few questions regarding the site navigation, online resources this program includes, and the program structure. So it looks like a few of you have made quite a bit of progress through the materials. A few of the top performers since we met earlier in the week are Mikhail and Elton. And I see a lot of you have taken time to fill out your profiles and updated with information. Let's take a look at one of those so I can show you some pretty neat features. I'm going to get out of the presentation here for just a second. I'll just show you my profile here briefly. So note on your profile that you can see your different groups that you're available in, different mentions and favorites, as well as your courses that you're involved in. And then within that, you can see your quizzes and assignments and different certificates. And then on your profile, you can see that you can add your name, and it would be really good if you could add your actual name as opposed to other types of online handles. This just helps us to identify you and kind of understand what's going on with stuff. Also note that you can add different social media things. So for instance, I added my Instagram, and you can see on here a little bit more about me. And this just kind of helps to build the community aspect that we're trying to build. Obviously later on, when you're done with the courses, we would like to feature your profiles perhaps. And so this kind of allows us to do that. Let's review some of the things that we discussed in our last session, starting from orientation. This part of the session will be presented as a quiz. You can enter your answers into the Zoom chat, and if you have questions, I'll address them as they come. So where would you go to find the courses and projects that are included in this program? Would you go to server.t-bio.info? Would you go to the program forum, the program syllabus, or the course page? So the courses, if you want to find out information about the courses and projects, you could go to the course page. And so I'm going to post the link to that in chat. Let's try another one. Where do you go with your questions about course material? Well, the answer to that is going to be the program forum. And this one's important to me specifically because it allows me to go in and kind of find out which questions you have. And then a lot of times that helps me to format my upcoming lectures so that we can go over materials that are more, I guess, targeted towards things that you want to learn. So the answer to that is the program forum. And here's a screenshot of that. So here's I guess another photo of the forum. And as you can see here, Beepsha has posted the video from last time session to your view. And I'm gonna also post a link to that as well. And you can bookmark these, so maybe it might make things a little bit easier for you to navigate. You can also leave comments and reviews on each of the courses. And this is also really good for us because it helps to, give people better insights into what's being discussed. And then also please leave a rating, and this also helps us as well. Let's try another one. So where do you go to run bioinformatics analysis pipelines? And I know some of you know this one because some of you are already running those pipelines. Do you run bioinformatics analysis pipelines in Microsoft Excel? Or do you log on to Google Drive? Or do you log on to Zoom? No, you know the answer is 1, server-t-bio.info. Good job, Eldon. So here's a photo of the server platform as it is, and there's several different sections that I want to go over just to kind of reiterate what they are. So the first one is going to be the sections by data type, that being next generation sequencing, mass spec, structural data, or others. Number 2 is where you can run analysis or play around with the demo pipelines. I encourage you to do so just so you can get a feel for the different sections. Number 3 is where you can learn about visualization, different types of statistical analysis, as well as machine learning. Number 4, if you go to the My Pipeline section, you can see the previous pipelines that you've run, as well as download those files and see which files you inputted. And then there's number 5, where you can see your account information. This is also where you go to log in. So if you ever go in and find out that you don't have access to something, make sure that you're logged in. Elton has asked if there's a number or a limit, or if there's a limit on the number of samples that you can run. I think Elia would probably be good for that question.
Sorry, what's the question again?
Is there a limit in terms of number of samples that we can run, that the students can run?
Oh, yeah. So the educational licenses that are included in the program registration are designed for the data sets that we already offer in the materials that are explained, for example, in the online lessons or the projects. Eventually, towards the end, we will show you how to leverage some data sets from the public domain. And there you will have to first submit the project proposal, and there you'll describe how many samples you are planning to analyze. But when you purchase a research license after you've learned how to use the platform, there's no limitation as to how many samples you run. There's a limitation of space, and you get 5 terabytes of space, which typically is enough for many samples. But it also depends on what you do with those samples, how many pipelines you run. So it's a little bit of a tricky question. The simple answer is right now you're limited more by the type of project rather than how many samples.
Okay. So our next question is, where do you go to see the list of sessions that are included for the program? And the answer for that is this syllabus. Sorry about that. They're testing the fire alarms in my building, so I had to mute my microphone for a second. But if you do go to the syllabus tab on the program page, you can see the different types of lessons that we're going to have and kind of be seeing what we're going to be doing in the upcoming sessions. So here's a screenshot of the program syllabus, and whenever you go to the program page, you can see that if you click menu, There's a drop down, and this is where you can access the syllabus as well as the resources, and then see the members and the faculty that are involved. And so that's right there. All right, so last time we met, Elia started to speak to you about cancer biology at the cellular and tissue levels. You learned about the roles of DNA changes and gene expression variation that's related to cancer risk, progression, and the outcomes. Let's revisit some of those concepts and make sure that those are clear as we move forward. So a doctor can detect cancer once clinical signs are evident from available data. These available data are going to be patient scans, x-rays, or different types of blood tests. Once they're detected, a tumor is going to be evaluated for malignancy. The criteria for malignancy includes increased cell proliferation, the loss of differentiation, some type of infiltrative growth, or metastasis to other organs. If the tumor that is performed from these cells has been is still contained within the tissue of the origin, then it's going to be called an in situ cancer. This type of cancer may remain contained indefinitely. If some cells experience additional mutations that allow the tumor to invade neighboring tissues and shed cells into the blood or lymph, the tumor is said to be malignant. So it's malignant whenever it goes to a different type of tissue. It's called in situ whenever it's in the same type of tissue. Malignant or escaped cells will establish, or they may establish, new tumors, which are called metastases, at other locations in the body. And the development of cancer cells is a process of deregulation of key molecular mechanisms. Cancer is a complex condition that's manifested in changes in cell regulation at many different levels. This results in an abnormal growth that we observe as what are called tumors. Tumors consist of cells whose growth and morphological characteristics are markedly different from those of normal cells. A malignant tumor develops across time and develops molecular changes as a result of DNA mutations. These types of mutations affect signaling pathways, which can be measured by changes in gene expression. So therefore, cancer is a disease of molecules and genes, and we even know how many of the molecules and genes are involved. These changes occur in cancer cells as well as its surrounding tissues, and this is called the microenvironment. This microenvironment includes many different kinds of cells, which include immune cells and cancer cells that learn to evade and even assist in processing the different types of things that occur in tumor development. So let's go through some things just to help you understand. I apologize, they're testing the fire alarms here. So to remember these aspects of cancer biology, there are different hallmark features that were identified. These are distinctive and complementary capabilities that enable tumor growth as well as metastatic dissemination. These have evolved over the past decade, and they now include sustaining proliferative signaling, evading growth suppressors, the activation and invasion of metastases, enabling replicate immortality, as well as inducing angiogenesis of energetic pathways, and then resisting cell death. There are an additional 4, which would be deregulating cellular bioenergetics, avoiding immune destruction, and then tumor-promoting inflammation. Last time we spoke about one of the most fundamental traits of cancer cells, which involves their ability to sustain chronic proliferation.
Joe, if you would like me to take over.
Yeah, Elia, if you don't mind, can you take over?
Okay. Okay, well, let me stop sharing your screen. Okay, sorry everyone for this change here. We're going to just quickly change screens. I'm going to share my screen. Okay, if you can hear, or if you can see my screen here. we're going to go through this, these hallmarks of cancer. And as Joe started mentioning, this also has been evolving over time. So what you see here on the screen are the 6 hallmark features of cancer that were originally established. And those are sustaining proliferative signaling, evading growth suppressors, activation, invasion, and metastasis, enabling replicative immortality, inducing angiogenesis energetics, and resisting cell death. So those are the traditionally kind of considered hallmarks of cancer. In the recent decade, additional 4 have been added, so there's a total of 10. And the additional 4 are deregulating cellular energetics, avoiding immune destruction, tumor promoting inflammation, genome instability, and mutation. So let's talk about these in detail. Last time, we spoke about the most fundamental traits of cancer cells that involves their ability to sustain chronic proliferation, right? So this one right here is sustaining proliferative signaling. And that is what drives tumor progression, right? So we talked how a normal cell becomes a cancerous tumor, is this sustained proliferative signaling. Normal tissues carefully control the production and release of growth-promoting signals that instruct entry into and progression through the cell growth and division cycle, thereby ensuring a homeostasis of cell number and thus maintenance of normal tissue architecture and function. Cancer cells, by deregulating these signals, become masters of their own destinies. The enabling signals are conveyed in large part by growth factors that bind cell surface receptors, typically containing intracellular tyrosine kinase domains. The latter proceed to emit signals via branched intracellular signaling pathways that regulate progression through the cell cycle as well as cell growth. So they increase in cell size and they divide more. Often, these signals influence yet another cell biological properties, such as cell survival and energy metabolism. Evading growth suppressors. So to prevent this from happening in normal cells, there are mechanisms to suppress this kind of growth. So cancer cells resist this inhibitory signal that might otherwise stop their growth. The majority pathways involved are autophagy and death cell receptor signaling, which is known as apoptosis, both of which can ultimately lead to cell death and reduction in tumor growth. Autophagy is a dynamic cellular recycling system that results in the degradation of cytoplasmic contents, abnormal protein aggregates, and excess or damaged organelles so that the building blocks, or amino acids, can be used to create new cellular components. Therefore, autophagy is a survival-promoting process. Cancers can upregulate autophagy to survive microenvironmental stress and to increase growth and aggressiveness. The autophagy process involves the formation of an autophagosome, which then fuses with lysosomes to form an autophagolysosome. The process is regulated by mTOR, MPK, PI3K, and MAPK pathways. So just remember these names and remember which process they're associated with. Activating invasion and metastasis. Cancer cells invade local tissue and spread. So what you see right here is the process of metastasis. And the process actually goes through the process of infiltration of other tissues. And so these are complex processes that leverage existing cellular mechanisms, such as the adherence junction signaling pathway, to enable invasion and migration of the tumor cells. Adherence junctions are dynamic structures that form, strengthen, and spread, degrade, and then reform as their associated proteins create connections with counterparts from adjacent cells. The adherence junction serves multiple functions, such as initiation and stabilization of cell-to-cell adhesion, regulation of the actin cytoskeleton, intracellular signaling, and transcriptional regulation. They can appear as bands encircling the cell, or as spots of attachment to the extracellular matrix, which are called adhesion plaques. Under normal conditions, this helps create order among the cells. Cell-to-cell junctions link cells to adjacent cells in a tissue, and they also regulate tissue homeostasis in critical cell processes that include tissue barrier function, cell proliferation, and migration. Defects in cell-to-cell junctions give rise to a wide range of tissue abnormalities that disrupt homeostasis and are common in genetic abnormalities and cancers. Enabling replicative immortality, cancer cells can revert to a pre-differentiated stem-like cell phenotype, allowing inhibited cellular division and other metabolic adaptations that enable survival in adverse conditions. Where there are multiple signaling pathways involved in these changes, two key components enable replicative immortality, Hippo and WNT. These are multiple pathways involved in this. There are multiple pathways involved in this characteristic of cancer cells. So each one of these processes has key genes and pathways associated with them. You will also see that multiple genes might have an effect on multiple pathways, and together they form these hallmarks of cancer. Inducing angiogenesis, which is what you see right here. Cancer cells stimulate the growth of blood vessels to supply nutrients to tumors. Angiogenesis is the formation of new blood vessels from pre-existing blood vessels. This plays an important role in tumor growth because it brings the necessary source materials and energy, and also eventually leads to metastasis because they infiltrate those and then eventually go through the blood to other locations in the body. Resisting cell death. One thing that we know about cancer cells is that they can resist death. They evade apoptosis, the mechanism that programs cell death once cell becomes damaged. Normally, apoptosis helps keep an organism healthy through growth and development, maintaining body tissue by removing infected or damaged cells. But cancer cells do not follow this process, no matter how abnormally they grow. The cancer cells may alter the mechanism that detect the changes or irregularities, preventing proper signaling and apoptosis activation. For example, apoptosis would start once there's a lot of damage in the DNA, right? And we know that the tumor growth and cancer development is associated with changes in the DNA, but nevertheless, they evade the apoptotic pathway and therefore continue to replicate with damaged DNA. So these are some of the hallmarks of cancer. Again, these are the 6 traditional ones, but today there are 4 more that are acknowledged, and some are given more priority than others. To study these processes, we can use next-generation sequencing data that reveals molecular changes that lead to deregulation of DNA, for example, mutations, and also show us how this process takes place. For example, changes in mRNA expression or alternative splicing. We also can detect changes in regulation of how one goes from the next. So remember that it goes from the coding sequence in DNA that is forming genes. Those genes are transcribed into mRNA, and eventually the mRNA is a product, a functional product, a protein, and those proteins can have different changes in them as a result of the original changes in the DNA, as a result of regulation of DNA expression or transcription, and also mRNA alternative splicing and other post-translational modifications that happen at the protein level. So when we speak about bioinformatics, we're going to be focused on studying the processes that we went over, these hallmark processes of cancer, relying on data. And so to study this data, we need to somehow use the sequences of reads that are generated in a sequencer machine, in a sequencing machine, to identify changes in DNA, find affected genes, and study mRNA transcription changes using some kind of analytical methods. Finally, we are also going to be talking, or we talked last time and we're going to continue talking today, how the data sources are different and typically used together to drive our understanding and knowledge about these processes. So we go all the way from cell lines, where we can perform high throughput experiments on different drug testing, for example, or understand the basic biology of what happens inside the cell, to put those cells in the context of a real system. They are typically implanted into animal models, and those are called patient-derived xenograft models, for example. Those could be mice with human tumors implanted in them. And then we also have clinical trials and different translational applications of the data and the information that is being collected. We have prepared a number of projects that could reveal some of those aspects that we just talked about. So let me briefly describe what these projects and data sets speak about. And if you're interested in any one of these data sets, you can let us know. Some of them are fully available on the site. Some of them are prepared as internal tutorials that are available in the programs. But all of the ones that I'm showing here are available for you as a part of this program. And you can let us know if one of those are going to be interested to you. DCIS chromosomes. So you remember the word in situ, which means contained in a tissue and not spreading beyond that tissue. There's a difference between early stage breast cancer and late stage breast cancer. Early stage breast cancer is contained within the tissue, and late stage starts going beyond one single tissue and spreading to other tissues. So how can we study the differences and whether we can predict how some early stage cancer can develop into a malignant tumor that is dangerous? We can look at DCIS exosomes. So exosomes are particles that contain RNA and DNA and mRNA, so different pieces of molecular material that is used by these cancer cells. And we can detect changes in the DNA, in the mRNA, and RNA-seq. And we can use that and then check whether the different features that we detect from exosomes can also help us detect and separate some more malignant or less malignant types of breast cancer from patients. Modeling cancer precision medicine is a project that we will do a session on during the program, but also it's available as a standalone project. And I saw actually some of you have been looking at that already. This project is actually going to focus on RNA-seq data from the original publication, but it will be used to understand molecular features or expression of genes that can help us identify subtypes of cancer and associate those subtypes with efficacy of different types of treatments, from chemotherapy to traditional chemotherapy to more targeted treatments that are being used in clinical trials. Changing response in cancer, immune response in cancer. Elton is saying he's interested in. Tumor-associated macrophages, or TAMs, are changing as a result of tumor development. And so we can study those different subtypes of these tumor-associated macrophages and understand how they differ between themselves, because that's actually the tumor recruiting these macrophages, which are a part of the immune response, to, instead of suppress the tumor growth, to support tumor growth and even metastasis. So we can study those molecular changes at the level of gene expression. And that's this project right here, immune response changes in cancer. We also have a project, patient-derived xenograft models. We'll be going through this in Transcriptomics 1 and 2. This is a study of the interaction between the animal models and the tumor, because we are looking at the microenvironment. And we would like to understand the changes in tumors between different, more aggressive or less aggressive subtypes, and the changes in the microenvironment that are maybe specific to those types of molecular subtypes. So Elton, we recorded your interest, so we'll be reaching out to you with some data sets from there, but you can also find these on the website. And if you need any help beyond that, we'll be happy to assist you with that. Okay, so Zainab, great, excited. So we will review a lot of these concepts throughout the program in future sessions as we do the practical steps of analysis and interpretation. A lot of the data sets from these projects and publications will be used. Are there any questions about what we covered last time and maybe added a little bit more information to that from today? Yes, please. Iftikhar, you had some questions. Please write those out in chat, or if you want to unmute yourself and ask those questions now, please go ahead.
Hello.
Yes.
Can you hear me?
Yes.
Yeah. Good evening. I think you said good evening.
Good morning or good evening for you. Yes.
Actually, I have some doubts about, because since I'm very new about this environment, it's really fascinating, and I'd like to thank Vipasa that she has given me the last video as well. I have some doubts regarding this. Whether these courses is payable, we have to pay again for these such courses like Omics.
You do not have. Yeah, once you've completed the registration for the program, these are going to be available to you as part of the program for free.
Okay, because I have seen a lot of omics of, epigenomics, metagenomics, bioinformatics, and all some courses there. So we say that $10 are the per license. Whether I have to pay for this particular program or it is altogether free. And second question with relation to that, it is an online, like we are having the online course right now. We are taking the classes. It will be like the same, or it's kind of videos. I have to go through these slides, and later on I have some questions I can interact with you. Please address these problems. Thank you very much.
Yes, so thank you for your question. As Joe was mentioning before, to find the courses that are a part of this program, you can actually go to this link. So there's a home page specifically for Bioinformatics for Precision Oncology program. This is the link. And in this link, now when you get there, you see down here, this menu is specific to this program. So if you go right here to courses, those courses are included. In other words, you don't have to pay for them. So these are online self-guided courses. What we do is we help monitor your progress and answer any questions that you might have. Where do you post questions? There's a forum right here. You can post any questions and we can address those questions on the forum. Or you can also request a meeting with us. Towards the end of the program, there will be a special portion of the program dedicated to independent projects. And that's where you can interact with us and ask any questions and schedule a meeting one-on-one. But for now, we will have these regular sessions like today, twice a week. And in those sessions, we will go through some of the material that we prepared for the program. But in parallel, you can go through some of the material that we have online, because a lot of what we will do in the sessions is going to supplement and add on to the material that is presented here online.
Yeah, got it. Okay, fine.
All right, great. So thank you for your question. Zainab, you have a question there. Okay, so let me read it out loud. Enabling replicative immortality, hippo signaling. Okay, so yes, we will actually go through some of the pathways. You can look right here, hippo signaling pathway, right? So these are pathways. The things I mentioned before are different pathways, WNT pathway, hippo pathways, And I'm mentioning these because we are going to be reviewing how these different pathways change as a result of looking at specific project data sets, right? So I wanted to establish the fact that we have genomic elements like a gene sequence encoded in the DNA. Then we talk about how that sequence is representing some kind of an expressed protein that has a function, but it's also part of a pathway of how they are regulated and what kind of is connected to what gene is connected to what gene. So we'll talk a lot about these different elements of pathways, functionality, sequences, and we want to connect these different things to what we know to be the drivers and characteristics of cancer, right? Because we want to understand the cancer biology before we jump into data and sequences and how to analyze it. So that's why I'm mentioning them. Now, if you want more information about the specific pathways, you will get the recording of this session, and I will also send you the slides. And you can go through some of the links that we will mention there and read some additional papers if you're interested to specifically focus on those pathways. But I wanted to just give that as an overview for everyone to kind of see how these things are related. Bipsha, if you could make sure that everybody else is muted for now. I believe Iftikhar is still unmuted, so we don't want to have that. Thank you. Okay. So are there any other questions about what we talked about today as a review, right? Before we go on into the new portion for today. Okay, if there are no additional questions, let's briefly review. We asked everybody to go through the introduction to bioinformatics, and some of you have done that. I do want to mention that the first lesson in Introduction to Bioinformatics is a survey that we wanted to use to learn about you and learn about your interests. And so we are thankful for those of you that have filled out that already, but we do ask that everybody completes this, gives you a good overview of where bioinformatics is now and other areas where it's applied beyond just cancer biology, but also in healthcare, and cancer biology is a part of that as well. So the course itself has 3 sections: Introduction to Big Data Bioinformatics, we'll talk about that today; Bioinformatics in Healthcare, we'll talk about that today; and Translational Bioinformatics, and we will also talk about that today. And so this material online really helps prepare you for what we're talking about today. And it's really short. It could be completed in under 2 hours of study. And again, it also provides us with some information about you so that we can continue and adapt our materials to your level and your background and your experience, as well as your objectives for this course. Introduction to Bioinformatics has a quiz at the end, and the quizzes actually help reinforce some of the materials that we go through to make sure that you understand the terminology, you understand how to use the information that is presented there, and also as a result of completing this material, this information gets updated on your profile, and we would like to help you get that profile to stand out as much as possible. We have a community of people like yourself. Some of them are, potential collaborators, researchers that you can collaborate with. Some of them potentially might be looking to hire a bioinformatician. Some of them might want to be hired, right? So it's a community. We hope to keep this community dynamic. And so we have a leaderboard where we highlight the top achievers from these online programs that we conduct to help share about the successes and the progress that our participants are making in these programs. So I encourage all of you to complete as many materials as possible. As we move on into more practical hands-on courses and materials, you will also get points for conducting, for running pipelines and conducting analysis. And that is actually where you can really stand out. You also get these certificates of completion, and that is something that you can post on LinkedIn. And of course, use it to demonstrate that you've completed some of these materials online. So today what we wanted to talk about is the history and significance of omics data and how it is changing what is happening in oncology. So medical data, or simply clinical data, is the type of information that have been used for decades in clinical use: temperature, heart rate, other data that is on the surface, right? So these are disease symptoms, and they are collected in what we know to be clinical data. But this data is on the surface. It is typically reported as a result of a patient feeling somewhere. Now, underneath, there are additional levels of regulation that cause this clinical symptom to appear. And the biomedical data is what is a combination of both clinical and this omics data that is underneath the surface. These include genomics, proteomics, and even the microbiome, right? So today we know how all of these areas are actually critical for the overall condition change from healthy to sick, and especially for tumor progression, as we have learned already. So a lot of these omics data types are driven by what's called high throughput sequencing, or also known as next generation sequencing. So let's talk about what is this data and how it has evolved rapidly over the course of the past several decades. Recall that it's been about 70 years since the structure and function of DNA was discovered. 50 years have been spent since that time sequencing the human genome, which originally took a very long time. And now, after 2015, the 1,000 Genome Project brought about a significant number of whole genome sequences to the research community. And in 2018, over 100,000 whole genomes were sequenced. Today, there's an ever-increasing number of data sets available that help researchers understand these subcellular processes and translate those into use by the clinical community. The technologies like next-generation sequencing are used to explore the roles, relationships, and actions of the various types of molecules that make up the cells of an organism. Omics data can be generated using next-generation sequencing. And to start learning about these types of data, the opportunities and challenges they come with, we will talk about how they are used in the clinical domain. Now, we'll talk about omics in general, omics coming from next-generation sequencing, but they really represent a variety of different types of data. So here you can see sequences of the DNA, which is called genomics, and that typically refers to changes in the genomic sequence. We'll talk about that more in our next lecture when we analyze and look for somatic and germline mutations. This also represents epigenetic data. Epigenetic data means changes, but that do not come from changes in the sequence of DNA, typically associated with effects of the environment. We'll also talk about gene expression data, which is the process of transcription of those gene sequences into mRNA sequences, and changes like alternative splicing. The significance of this data is increasing as we get closer and closer to translating the information obtained from research projects to actual clinical use of next-generation sequencing data. The way it has been happening over the past decade is changing the way we can predict risk, identify a prognosis of disease, response to treatment, stratification of patients by molecular subtypes, drug repurposing, diagnostics, and even target discovery. In fact, many aspects of NGS have already changed both the understanding of the human genomic changes, for example, tumor development, or agents that cause such changes, for example, pathogens and viruses. It has also changed the way that we can treat patients with targeted, safer, and more effective treatments. So here you can see some illustrations by clinical research centers of how they are translating genomic medicine into practice. You can see here expanded definition of self, right? So we can better understand the germline genome, the tumor genome, the microbiome, and the different infectious agents that interfere with some of these processes. We can diagnose conditions early on, hopefully at conception, preconception, and even soon after birth. And also know what the effects of different changes are going to be as we grow older. Earlier disease detection before clinical manifestation of those disease symptoms. And then we also, this is changing the way that we can do treatment of disease and prevention. So for example, Herceptin is a drug, an antibody-based drug that targets the HER2 protein. We'll talk about that more later. We will also talk about, for example, hepatitis C, a major cause of liver cancer. And we'll talk about how can we detect presence of viruses in the human cells and how that can affect our ability to prevent the spread of cancer, for example. And obviously drug safety and really optimization of that treatment as we now have additional sources of data that are non-omics, but nevertheless generated by the patient. Now at the center of this process, what you can see right here, this is from the Center for Cancer Research in Australia. You can see the process is illustrated by the clinics interacting with the patients and sending data to the labs, where a central role of bioinformaticians is to process and analyze that data that is then passed on for curation and interpretation by the tumor board, for example, and eventually translated into a treatment plan. So the role of bioinformatics is central to the process of translating a lot of the omics data information into clinical use. And a lot of that process has been driven by a reduction in the cost of sequencing, of next generation sequencing. So cheaper, faster, and more efficient sequencing that started with shotgun sequencing that was introduced during the Human Genome Project, but has significantly reduced in price very fast from when it was in 2001, the cost was 100 million to sequence the whole human genome. And then today, the cost of a whole genome sequencing is about just over $1,000. Now, why is it important, this price point of $1,000 or below? Well, because we can compare that price to some of the other standard tests that are being done in the clinic. And also we can see how much data, how much information these other tests are generating. So for example, X-ray, right? It's about 30 megabytes per image. If it's a high resolution image, has a lot of detail in it, and it costs about $250. And it's about 30 megabytes in size. Computed tomography, which shows, for example, helps detect tumors in the brain or inside the body deeper than what we can see, about $800, and you can see between 3 and 50 megabytes. And then nuclear medicine, about $636 and about 300 megabytes in size. What is the size of a whole genome in terms of the raw data that is processed from the whole genome? Well, the cost is just over $1,000, and the size is several gigabytes, right? So depending on the depth, it could be anywhere between 2 and 10 gigabytes in size. So much larger in terms of data size for a price that is somewhat similar, right? So if we just look at those two aspects. Now, what's more interesting even than that is that just recently, just in 2020, this is from the MIT Technology Review, China's BGI said that it can sequence a whole genome for just $100. And so watch this video. Sorry. Let's watch this video real quick. The reason why this can be achieved, this price point can be achieved, is because of this, because this is fully automated, right? And so that really becomes a question of automation, and high throughput means that we can generate a lot of data per single sample, and the process is automated. Now let's take a look at what is this next generation sequencing data. The next generation sequencing technology allows for advanced studies of genomic mutations, gene expression, metagenomic data, because it captures a snapshot of targeted genomic regions, whole genomes, or the whole transcriptome. Whole genome, whole exome, or whole transcriptome sequencing provides a comprehensive view of a cellular genome, the changes associated with that genome, the expressed portion of that genome, for example, whole exome sequencing, or provides us with a detailed view of the transcriptional profile at a given biological moment. These methods greatly enhance the power of discovery when we look for changes associated with conditions like cancer. However, to study this data, we have to process the raw reads and turn them into structured data that is ready for analysis. To understand this process, we need to look at the process of next generation sequencing from the preparation of the sample all the way until we have a readout of a single read. So what it does, and next generation sequencing technology captures DNA sequences, generates digital data from those samples of DNA. So first, RNA or DNA is extracted. It is shared or broken up into small pieces. Those have adapters attached to them, which is called the library preparation step. When they are applied to a flow cell, amplified inside the flow cell, and then sequenced to detect by synthesis with reversible terminators to detect which letter is seen at a given position inside that short segment. So as a result, we have a sequence of C, G, T, and A letters, nucleotides, and we'll talk more about what those nucleotides are. They're all going to be in cDNA format, so these are going to be DNA letters and not RNA. And those reads, these kinds of segments, can be anywhere between 30 and 300 base pairs long. One major challenge when we have such easy and increasingly cost-effective access to data is the ability to integrate that data to understand what is happening from a heterogeneous big data problem, right? So it becomes a problem of big data. There's too much data compared to, for example, clinical data. And extracting information from that data requires looking at heterogeneous sources with different types of information that speak about the same biological condition that we would like to investigate. So because of this, let us turn now to the big data challenges and opportunities. Because big data is both a problem and an opportunity, and it requires a certain appreciation of the properties of this kind of data for us to be effectively using it to generate meaningful information. Now, from a biologist perspective, we know that we can observe specific changes at the phenotype level, and we would like to understand how those changes are associated with what we understand to be the molecular process that results in these changes. And a lot of times we were trained to look at those observations, to then ask a biological question, to then generate a hypothesis. That hypothesis drives us to create a controlled experiment where we generate data and collect that data, develop an analysis that helps us, for example, prove or disprove a hypothesis. And as a result, we have a report, which we call information, right? So we go through this process to arrive at some kind of a result. Now, typically, we think of a result as a logical connection between different elements that we can study, right? So oftentimes we hope to arrive at a clear association between different genes that are combined into pathways, right? So here we have a pathway, for example, and we study hallmark capabilities, right? So we talked about these hallmark aspects or characteristics of cancer. We know that they are driven by changes in gene expression. What genes? Well, here we can split those genes into different types of circuits or pathways that interact with each other and drive a certain condition. When it comes to big data, when we have ability to study all of the changes at a given moment in time from a sample, this equation changes. Essentially, it looks more something like this. We have a lot of different players. Those players have complex interactions between them. And it's really a challenge to reduce everything to the type of a hypothesis testing experiment that we typically expect to generate information. So that is what's called a big data problem. Now, what makes data big data? First of all, there are several V's, as you can see right here, that characterize big data. And those are volume, variety, value, veracity, and velocity. Volume refers to the amount of data, which is huge. For example, RNA-seq yields information about tens of thousands of genes. Or if we talk about genomic data, we're talking about millions of single nucleotide mutation variants that are possible from a given sample. Variety. This data is unstructured and can be assembled in many different ways. Oftentimes, the same phenomenon can be described in many different ways depending on the type of sequencer, preparation, library preparation techniques, or even handling that affects what we see as a result of that process. Value, information that we can extract from this kind of data is dependent on many aspects that include noise in the data, technique error rates, and completeness of information a single sample can contain. Veracity, some samples have low coverage, others have high coverage, but not full sequences. Others might be generated by an older sequencer that had a high error rate. Even so, including all of this data together can complete our picture of a specific phenomenon that we are studying. And finally, velocity. Another important aspect is the change in data types and new information that is generated. For example, Sanger sequencing was replaced by shotgun sequencing, and eventually now we have an explosion of single-cell sequencing, and then we have new types of sequencing techniques like Oxford Nanopore. So we're always learning new things from these new additional types of data that are coming, and we have to adapt the way we study these processes to incorporate these types of new information that are coming from these new techniques. Now, the high throughput sequencing, you can see that it has really significantly grown over the course of the past 20 to 30 years. So back in 1989, first sequences were started to be assembled by NCBI, the National Center for Biotechnology Information. And the rate of growth of this data has significantly risen over the past couple of decades. So you can see also how this has impacted the ability to translate those logical schemes that we are used to, that are very logical and easy to understand from a pathologist or an oncologist or a biologist perspective. Now, even those people have to deal with this influx of available data and information that is unstructured and changing and expanding all the time. And so as a result, we have to change the way that we deal with this data from testing a hypothesis and extracting information to adapting to the constant flow and expansion and the complexity of data that comes out. For example, the TCGA, the Cancer Genome Atlas, has 33 different tumor types, over 11,000 patients and growing, and 7 different data types. And you can see the growth of that data just over the course of the past decade is expanding, and more publications are using the data that is generated to propose new ideas, to propose new characteristics associated with cancer. As a result, many researchers are turning to data mining. Data mining is our ability to detect patterns in big data that has complexity. So here you can see, for example, analysis of different types of cells. And here you can see the patterns that those cells have, right? So these are complex patterns that are not going to fit standard statistical approaches, for example, of hypothesis testing, that expect certain data distribution and properties. When we talked about low throughput data, that low throughput data was easy to convert to information. So here you can see, for example, weight and size, two features can clearly separate men and women, right? And that is easy to do by traditional methods of dealing with this kind of data. With this kind of data, that becomes more and more challenging because the patterns are complex, and we have to rely on data mining techniques, which we will learn about. Those include things like clustering, dimensionality reduction, but also require a lot of pre-processing steps because they work only on structured data. There's also a growing interest and use of machine learning. And so that's another topic that we will talk about in the context of this program. Machine learning, as you see, became more and more increasingly popular after the human genome was published, after the year 2001. And now it is in many publications becoming more and more commonplace. And so the role of machine learning is really a result of the growing amount of data that is available and that is being generated. And therefore, we have to also understand how these machine learning methods work and what do they really do to the data when we feed some black box, right? So that is another topic that we will discuss throughout this program. As a result, there's a new process to dealing and Zainab, yes, we will definitely give you a brief introduction to machine learning. In fact, as you get through transcriptomics courses and get to transcriptomics 3, we'll start talking about, we're not going to jump there right away, but we will have specific sessions dedicated to machine learning. So the process of that interaction, right? So before that, I was mentioning how there's a connection between people generating the data and people using the data that goes through a central point of bioinformaticians that have to analyze, curate, and interpret the data. And that process is becoming more and more dynamic, right? We have to speak the same language for that process to work. But what it actually does is that someone else, right, a person at the clinic, a biologist, a researcher, has to specify a problem, and especially they have to specify that problem from the point of view of data. What kind of data are we going to generate? What is that experiment going to generate? How are we going to extract the answer to the problem from the data that is being generated? Can we find that data from previous experiments that were already conducted or from publicly available data that is already out there? That's why we have to build analytical tools and environments that integrate databases of available experimental design. Oftentimes, we generate some kind of a model based on that data. The model could be simulated, the model could be predictive, but that model essentially helps us analyze and visualize data in such a way that we can make decisions and interpret the results. And so this iterative process is what you will see more and more as the role of a bioinformatician to facilitate the discovery and the translation of information found in these complex data sets to a practical use, either in discovery, in research, in basic research, translational research, or in the clinic. In real life, it might look like an integration of these different aspects of cancer biology that could be studied on animal models, on patients, and on single cell lines or just cell lines. So this problem of big data is really changing the role of bioinformatics. It's also changing the way that other people that traditionally have not had to rely on bioinformatics have to now utilize bioinformatics in every area of their research or even clinical practice. So just as an example, I wanted to highlight, for example, the evolution of breast cancer classification that we will be talking about very soon. The evolution of breast cancer classification, right? We talked about the hallmarks of cancer, but those hallmarks are just general attributes, characteristics. And today, when we apply these different characteristics to study what happens in the real world, what happens in real patients, we have to understand how the molecular data is changing our ability to separate those different cancers into groups, into different defined molecular subtypes. We will also talk about how we can integrate the different data types and what they allow us to see to study a variety of things that we cannot expect from patients, like, for example, this project on efficacy of drugs, which obviously is not going to be possible if we just have access to patient data. Any questions on what we had for today's session? These couple of first sessions are really introductory to get everybody on the same page. And so that's why we spent some time last time and today to speak about the learning environment and the process that we will follow as these programs develop and become more and more practical. So I wanted to make sure that everybody is familiar with a little bit of cancer biology and a little bit of the problems associated with next generation sequencing and big data before we jump into the practical parts of the projects and the analysis. Now, great question from Iftikhar. How to get the NGS data and to understand or interpret it. So we will first of all provide you with some examples to get started with. In fact, next session is going to be dedicated to identification of DNA variants, so single nucleotide variants based on analysis of genomic and exome-seq data, where we will talk about germline and somatic mutations, and what are some of the practical steps like mapping and detection of variants and comparing between the normal and the tumor data, and we'll actually use a real data set. And then how do we actually interpret that kind of information, right? So what are the file types? How do we interpret those file types? How to visualize that data and things like that. We'll do that next time when we meet on March 16th. And after that, we will also talk about RNA-seq. How can we study gene expression? And eventually we'll learn how this data can inform us about these different molecular processes that we started talking about that define cancer biology. Okay, are there any other questions, Darko? Thank you for additional information. Yes. Where to get the data? So we will have a session on how, let me actually go through, there are a lot of questions about some of the things that we actually plan to go through in this program. So let me kind of describe what will be covered so that we can stay focused. So there are other, repositories for the different things that we will do. But if you go right here, under syllabus, you can see what are the different topics that will be covered. So we already did Introduction to Oncology, where we talked about cancer biology challenges. We talked today about big data bioinformatics and some of the things that we have to think about. Next time when we meet, we will talk about DNA variants and mutations, and so how to analyze genomic data. Then we'll talk about next generation sequencing transcriptomics for RNA-seq data. We'll talk about statistical methods of analysis that are used for analysis of RNA-seq process data, tables of expression. We'll talk about interpretation using gene ontologies, functional, and gene set enrichment analysis based on pathways. Then we will actually get to where to find data and how can we use some examples from cell line data and from animal models. And that is data found on NCBI, which is the National Center for Biotechnology Information. You can find a lot of different information there. So please visit the link that Mohit gave. That explains where we can find RNA-seq data on cancer. Then we'll talk about TCGA, the Cancer Genome Atlas, where we can find data from patients, and there you'll find data such as clinical data. I'm not sure you can get MRI or X-ray or something like that from patients without being approved first, but you can go through the approval process. We'll also talk about different databases of mutations like NCI and COSMIC. Then we'll start talking about projects, right? So our goal in this program is to prepare you for independent analysis of these kinds of data so that you can actually think about how to apply the techniques that you've learned about to real projects, right? And we'll not only talk about where to find the specific data set, but we'll also talk about how to select that data set and how to propose an idea that is valuable and competitive so that your inquiry and translational value of the analysis is going to be solid. For example, how many samples to include, and reproducible. So how do you record all of the steps that you do so that others can arrive at the same conclusions as you do? And then we'll spend some time going through project examples, how to do classification of subtypes in breast cancer, how to deal with different cell lines, and then we'll also include liver cancer from TCGA and analysis of the different factors that we have. I believe, yeah, this is just a problem with the image. So that's what we plan to cover. I hope that you can join us for every one of these sessions. And if not, the recordings are going to be available. So you can already see the recording of the previous session that was posted right here on the forum. If you want to find any of the previous recordings, please make sure to go there. There's going to be a post from Beepsha where we have session recordings. And so you'll see here the different recorded sessions with links to YouTube. All right, so great. Thank you everyone for joining today. We ran a little bit over time, but hopefully you enjoyed the session. And we'll be looking forward to seeing you again on May 16th next time. Thank you, Darko and Joe and Mohit and Bipsha for joining today also. Okay, what do we need to prepare for next week? Again, if you have not finished Introduction to Bioinformatics, please complete that. Most importantly, complete the surveys that are included there. And start going through Introduction to Genomics and Genomics 1, depending on how much you can achieve before now and next time. Those are the two next courses that we are going to be discussing, genomic data. All right, have a great rest of the day. Take care.